Good morning. It's 9 o'clock here in San Diego, so we will begin the webinar. Thank you very much for your support of the webinars, and thank you for joining us today. Today's webinar is on chemical searching, clustering, and chemical analysis using the ICM Chemist software. Just as an introduction, my name is Andrew Uri, and if you have any questions after the webinar, please feel free to email or call. During the webinar, please use the text messaging panel to ask questions, and I'll try to get to them maybe during the webinar or at the end. And you should have received information about how to receive a 30-day ICM Chemist license, so you can follow along with the examples shown in the webinar. And in the same email, you should have received also the files that we were using today. You should have this link, and you can just download it and unzip the files. There were some people that requested licenses last night, and so I will send them later today if you have not received them yet. So today's webinar topics are how to plot chemical data, XY plots, histograms, how to generate curves and heat maps, and then how to perform chemical clustering, so you can have a large database of chemicals and then cluster them and select a subset of those, and you have a tree. And then how to search a chemical database. So you can search by a variety of different queries, which I will show, and how you can filter those queries and process them. And then we'll end by looking at our maximum common substructure method that we have, which is a useful tool to look at chemical similarity and prediction of activity. So we will start by opening ICM Chemist, and you should see a window like this. And earlier in the previous webinar, we showed some searching. And so this table here is a chemical spreadsheet that is delivered when you go to the search tab and you go to the Chemball, and I searched cyclooxygenase 2, and it delivered this spreadsheet. If you have the files that you downloaded, you can go File, Open, and then STF, and you can open this file. I also added some properties to this table. So we have the cyclooxygenase. These are all chemicals that have been annotated as cyclooxygenase binders in Chemball. So I've also added some additional properties to this, including molecular weight, log P, solubility, polar surface area, molecular volume, and some other rotatable bonds and hydrogen bond acceptors. So some Lipinski rule stuff. So the way I did that was just to go to chemistry and then calculate properties, and you just select which properties you want to predict and do that. So we have this data. So it might be useful to be able to plot this data. I showed briefly last week, 2 weeks ago, how to do that, but I'm going to go into a little bit more detail about how to use plots. So on the right hand side of the chemical table, you have a button here which is a plot button. So we click on the plot button here, and it's asking us initially for our initial X and Y data. So I'm going to choose molecular weight for the X-axis and prediction of log P for the Y-axis. And if you just go OK, you get the plot on the right hand side of the screen. You can drag it here to make it bigger. And as we did the other time, you can add additional levels of detail to this this plot. We can right-click on the plot again and say Options, and we can plot additional data from here onto this plot. So we could say we want the size of the points to be related to molecular area. So we go to this section here is all related to the points, to these dots. So you can it's going to add this anti-aliasing, makes the dots a bit more rounder. But so the points we have the size. So the size is going to be related to molecular area, and you could change the shape if we wanted to, but we're not going to in this example. You can add point labels. You can say I want each point to be labeled by the UniProt ID or something like that, and the label size. And then we have the option for color. So we can say we want to color by solubility. So we drop down here. So all these options here are related to whatever you have in your chemical table. So we're going to choose solubility, go apply, and you can see now it's colored by solubility from red to purple here. We can change the coloring if we wanted to by clicking on this edit button here, and then you can change the gradient by dragging this gradient bar here, or you can color by a fixed range, so from 0 to 1. It's not so colorful, but and if you want to go back, we just click on that button here, and we can calculate. We're going to calculate the coloring from the data that's provided, not in the fixed range. So go OK and apply. Okay, so we have our plot. So you may want to change the label, the labels. We can go to the shapes, for example, and say we want to have a different shape for each UniProt ID. We have some different species in here. If you do that, that adds another legend on the right hand side that you can relate to different. So circles are human, triangles are sheep, mouse, and rat, what have you, like that. There are some additional options on the bottom side. Anti-aliasing improves the graphics representation of each sphere, so they're nicely spherical, and you can check that on and off. You can also uncheck the axes and just have a blank screen, and you can also remove the grid as well. So you can just have a display like this. I'm going to add those back. Okay, so as we showed a couple of weeks ago, this is fully interactive, this plot. So if we make a selection here, for example, we can find this chemical in this plot by right-clicking on here and say center selection. This is our molecule. You can also do it the other way. So we can see that we can make a selection. We can zoom in and out of the plot by using the middle mouse button, and you can use the left mouse to make a selection. This makes a selection here with red crosses, red boxes, and the selection is propagated to the table shown here. As we're investigating this plot, it might be useful to be able to hover over a particular point in the plot and see more information about it. So at the moment you can go here and then find it in the table, but you may want to just browse the plot. So you can right-click again on the plot, choose Options, and then we have this option here in the data saying balloon. So if I write, so basically it'll have a little balloon window come up related to any data in your table. So if you wanted to display the 2D sketch, you just type mole here, and go apply, go OK. So that means now if we hover, I'm not sure if it projects across the GoToMeeting, but it should do. If you hover over, yeah. So if you hover over a point, you can see the 2D sketch of the chemical. So you can also add other data. So we could go mole, then add comma, and mole PSA or something, polar surface area, and then apply, go OK. So now if we hover over a particular point, we should have the polar surface area also displayed in the window. So for zooming, you can use the middle mouse and globally zoom, or you can use the middle mouse hovering over an axis, so the x-axis here, and you can zoom in on the x-axis shown here, or on the y-axis shown here. You hover over. And also if you hover or right click over on an axis, you get additional details, so options that you can choose. So you can right click here and you can choose x-axis options. So you can change the logarithmic scale if you wanted to, or you can change the range and the steps for each point, and you can modify that. And it's the same in the y-axis as well. So you may also want to annotate your plot. So you can make it. We can say we're interested in these, maybe this region here. It's sort of the Lipinski rule, log p below 5, and molecular weight below 500. So we can maybe annotate this for our colleagues. We can right click and choose annotate, and then you have some options to add some annotation. So we can say we can draw an ellipse over here, and you have the fill color. You can change the fill color here, and the outline color here. Change the size of the lines. You also add text just by clicking on the so this, to get this window, you right click, you say annotate, then you can add some text here. So, and then you can also add lines and what have you. Then you can delete whatever you've done by just selecting them here. Or you can export as a picture, an image. You can right click, choose save as image, or copy the image to clipboard as well. Okay, I'm just going to delete that annotation. Okay. So you may also want to filter this plot. So we can do that by clicking on the filter button next to the plot. So we just if we double click on a tab, it loads both of them at the same time, so you can visualize both on the same screen. So if we wanted to change the molecular weight, we can see it, the plot will dynamically change. Use this dragging bar if you bring up to 200 molecular weight and below 500. It's changing. We can see in the column here that in the table that this column is filtered. We have this little filtering option here, and the same with log p. We could do that. We could bring this down to 5 like this. You can also have multiple plots, so we can add another plot, and let's go OK. And then we can just double click and it shifts them around the screen, and like this. So you can have multiple plots which are all fully interactive with the table. So you can select and drag. You see the fully they selecting in all tables, all plots, and all in the table. I had a question, actually, quite a good question. Can you use rather than a dot, can you use the 2D image of the ligand as a point? I'm not sure if that's possible. I think it might be possible in the future. It was something that has been working on, but we could try. Okay. It might be possible. I'll get back to you about that. I have seen something along those lines. So yeah, it would be nice to be able to have the sketch, I think, as a dot, an image of this as a dot on here. We can maybe look at that towards the end. Okay, so another plot you may want to do is a histogram. So you can select a column that you want to prepare the histogram for, and then you can right click and you can choose column, right click on the column header and choose column histogram, and then you have the histogram here. So we may be interested in some chemicals we can make a selection in the histogram. See, it's selected here. It's selected in all our other plots that we have. Here. So we can we have a selection in the table. We can extract a selection that's made. So we can right click on a selection and say copy. So copy selection to table new. Sorry, but we just give it a name. So we have a new table. Just going to delete that actually. They just copied one column because we have a cross section here. We've got this column selected. So I'm just going to unselect this column, and now it will make it this selection again. We can just right click and say copy selection table new, and we have a new table just with our selected residues. And you can also build a heat map of this data. So we can say I want to see a heat map of this selection I just made, molecular weight, log P, polar surface area. So I went to the button here for plotting, went to the heat map tab, and you can add these different options here. Let's go OK, and you can see it plots the heat map on the right hand side. Shown here. And another kind of plot you can do is a pie chart. So similar thing. I've just got two tables here. We can click on the button again for plots. We could just go to so we could just be interested in the uniprot ID. So we could just plot what kind of species that we have in our data. Just go pie, and that will go OK, and then you have a fully interactive pie chart as well, which is so you can select in here. Okay, so that's for the plotting. Now we have. I'm just going to delete this table. I don't need it anymore. Going to this hammer button here expands the panel, so you can close it or you can open it, whatever. So we're going to move on to chemical searching. So 2 weeks ago we showed some ways, some cyclooxygenase inhibitors, some famous ones like Vioxx and Celebrex, and we're going to see if we can find those chemicals using chemical searching in our database here of chemicals from Chemble that are also cyclooxygenase 2 inhibitors. You can see I have some columns already filtered, so I'm just going to remove that filtering. So right click on the column header and say filter clear all. That clears all the filters. So we have a table of 5,713 rows of chemicals. And so we're going to use some of the chemical search options to find some well-known cyclooxygenase drugs in this table. So to perform chemical searching, we have two buttons here. This one on the right-hand side is for editing. This is for drawing, which we showed last week. You can sketch molecules. But the button next to it, which is a ring with a pair of binoculars, you can click on that, and it opens a similar panel, but also has this extra panel here for search on the right hand side. So we know that some of the cyclooxygenase inhibitors that the drugs that we drew last week, they had a sulfonamide group within them, and so well, basically you can use this to sketch. So you can draw a substructure and then you just go search and it will try and find this ring. But in this example, we're going to start with sketching a sulfonamide group. So we can use you can use your keyboard, or you can use the templates on the right hand side. You've got the sulfur double bond here. And we're starting with our first search query. Oh, like this. So it's a sulfonamide. So using the keyboard to add the annotation. This is nitrogen. And so we could say that we want to find chemicals that have the sulfonamide group. So we can say we want to set this atom here to be an attachment point to the rest of the chemical. So we can right click on here on the atom. It brings up this sub menu, and we can say we want this atom to be an attachment point. So hopefully GoToMeeting projects that. You can see that there's an asterisk here now, which means this atom is going to be bound to another bond and the rest of the chemical. We can also be more specific. We can say we know that the Vioxx, for example, has a ring here. So we can right click on the atom and say we also want this to be an attachment point, but we want it to be this atom to be part of one ring. So we go ring, we right click on the atom again and go down and choose R1, and we have our first query. So it's asking us to tell us what our query input is. Our query input is our sketch. It's the draw, so we're okay. The table that we're searching is this Chemical COX-2 inhibitors that we've downloaded previously. You could also search a file on your computer, or there's another product Molsoft has, which is a chemical cartridge called Molcart, but it's a different subject altogether. We won't do that, but you could have a file and search that file. Particularly useful if the database you're searching is very large, so you don't be able to read it into the computer. You can just have it on there. Then we have the move down, and we're saying query up, and you also got the column that you're going to search. You're going to search the mole column of the table. So now we have the query types. You can search by substructure, which is what we're going to do, fingerprint similarity, or exact match. If we had fingerprint, we have a distance here, and we have also options for matching stereo, ignoring the salt if there's a salt in your table. You can also search tautomers and other options for maximum number of hits. So we can say then you can define how you want your output in the results part. So you can say I just want ICM to count how many hits I have, which is sometimes useful if you're trying to bring the database down to a smaller number. You can say selecting source. That will mean that all the hits will be highlighted in blue, and then you can extract it to a new table. But in this example, I'm just going to save results to a new chemical table called result. You can save it to a file if you wanted to. And so you just go search. Okay. So the search is done. And you see now we have two tables. We have the original Chemical COX-2, and then we have the new one called result. And we can see that we found the sulfonamide, which is good. But we're also finding chemicals such as this one, where the sulfonamide where it's actually linked between two different substituents. And we know that the drugs that we were looking at last week were had like a sulfonamide group. They weren't between two substituents. So here, for example, we have a phenyl and then a thiazole, and so we need to. We don't really need that. We want to change our query to be more specific, to have like a terminal sulfonamide. You understand that? So if we go back to the query, we have the sketch here. We don't want this. What we're trying to do in this next query is to say we don't want this nitrogen to be connected to another bond. We just want it as drawn. So we can say right-click on the nitrogen and then choose connectivity and choose as drawn. You could say I want this nitrogen to be connected to a number of other atoms, but we don't. I'm just going to say as drawn. So then you have this this new search criteria. So by changing the connectivity to nitrogen, we're going to find only terminal sulfonamide groups such as Celebrex, which is the drug, one of the drugs we sketched a couple of weeks ago. So now if we go search, and sorry, for some reason we had a little bit of a crash there. It's good, okay. And so I can get back to my query, sorry, the search query, and then let's draw that sulfonamide group again, and it's going to go we're going to say nitrogen, and this one is going to be it's going to be an attachment point. It's going to be a ring membership R1. This nitrogen is going to be, this is what we were just doing, connectivity as drawn. And so now we should find drugs such as Celebrex. So if we go search, it's going to, yeah, okay, yeah. So somewhere down here, so now we have a new table result, and the table has 595 rows. So we're gradually narrowing down our search. So we have 595. Somewhere in here will be our Celebrex, and we can so Vioxx doesn't have a Celebrex has a sulfonamide. Vioxx has a sulfonyl group. So we can change our query again by also saying, by editing the element here as well. So rather than, we want to find sulfonamides as well as sulfonyls. So we can right click on the atom here and go to element and go to custom, and now we can just say we want a nitrogen and then add a comma and a carbon, and we'll have both of those, they'll both be considered in the search. And we can just change the name of the result table to result 1 and go search. Yeah, and this finds sulfonyls as well as sulfonamides. And so it's going to be slightly larger. There's quite a few of them because we got a dual search. So somewhere in here we have our drugs. So I'm just going to delete these results. I'm just going to read in the drugs that we sketched last time. We go file, and then I've got, it's called COX-2 drugs. We have these 4 inhibitors, four drugs, Vioxx. You see with the sulfonyl, and Celebrex with the sulfonamide group shown here. So we can actually just take this table and search our chemical results as well. So we can go to that chemical search window again with the ring and the binoculars, and rather than sketching, we can choose query input, and we can choose the table. So we say we want to search these COX-2 drugs. So if we go there, they're displayed in the search, and so we can try and find. So it's going to search these chemicals against the larger chemical database. So just we do search and go search, and we should get a result. We have a new table called results here. So we found some of those plus some other ones. Okay, in addition to when you're searching, you can also add some other additional filters, such as some conditions. So if you right click in this panel here, we can add a condition and say we want the molecular weight to be less than 500. Add another condition, we want log P to be less than 5. And add another condition, just double click and then you get the, so you can say the number of hydrogen bond acceptors. So shown here, and then you can say less than 10, for example. So you have your inbuilt rules that you can add here. And that's going to filter the hits that you get from searching. So you just go search, and it'll do the same thing. So you could count hits. You'll find we have 72 hits with these drugs and these. You can also compare chemical tables. So if you read in the, I'm just going to delete the results again just for clarity. So if we double click, it separates the two tables, Cox drugs and our chemical data. If we wanted to compare this table with this one to try and find it, we can also go to chemistry and choose compare two sets. So we have, it asks us what's the first table. First table is chemical COX-2. Second table is the COX-2 drugs. We can do an exact match. We go OK, and it will tell us that it's found these 5. You've got one selected here. If you have a very large table and you have selections that you want to see them on their own, you can right click and choose show only selected rows, and there you can see we have found the four drugs. So then you go row selection, and just below here you've got show all rows, and I'll go back to the original table. So in the chemistry menu there's some other options, such as select duplicates. So it will go to a table and it will flag those that are exactly the same, and you can delete the duplicates. You can also merge two tables depending on certain criteria in each. So you need to have a common column between the two tables, and then it will merge those two tables. And also, as we showed last time, in the file, tools table, there are some other options such as append rows, add external columns, and pivot, and other analysis as well here for tables. Okay, so that's chemical search. So we're going to move on to some chemical clustering and also looking at maximum common substructure. Just wanted to show a couple of slides. So in this example, we're going to read in a database of drugs, and we're going to just basically use the tree option. So we're going to cluster them basically, so you can select a subset of similar chemicals. But also, you're going to introduce this option of maximum common substructure. So maximum common substructure is it's a metric for chemical similarity. So it can be used for searching and activity predictions. So the maximum common substructure, or MCS, of two compounds is the largest substructure that appears in both structures. So using MCS is a useful measure, because you would assume that the largest common substructure of structure-related drugs is likely to be a component of their activity. So it's useful to be able to see, what is the core. So we can make a tree, and then we can see that these ones all have this common substructure here. So so you can use it to look at the common core, and it also can be used to visualize using a dendrogram, which I'll show in the next slide, like this. So you have a big table, you can analyze the maximum common substructure, and then you can zoom in and investigate this dendrogram, and you can see which chemicals are connected to. So you're matching, you're graphing the maximum common substructure between the chemicals. Okay, so we're going to start first of all by we're using the drug database that we have in ICM. I'm going to do edit delete all. And we'll go file. In your distribution, and if you've downloaded ICM Chemist, you should have this file. It's called dictionary. It's a dictionary of drugs, basically. So to cluster this table, we're just going to add some additional properties to this spreadsheet. So I'm going to go to chemistry and then calculate properties. I just choose the ones we did before, molecular weight, log P. Let's go OK. So they're predicted here. So we can build a tree of the clustering tree. So we can click on the clustering here, and this button here. Then we get this dialog window. You can give a name for the tree if you wanted to. It's going to take by default the names from the. It's going to label the tree by the name of each compound. So if we were going to do the maximum common substructure, we would click here. It takes a little bit longer to run, so I'm not going to do it. I have an example already prepared. And then we have, what are we going to cluster? You can cluster by any value or chemical, basically. And you can use fingerprints, which is the default option. So we're going to use that, and we're just going to go OK. There's some additional clustering methods that you can play around with, but by default we use this one. And we just go OK, and that will deliver the tree for this. So this is a database of drugs. So now it's colored the table according to the tree. So if we scroll up and down, we can see different coloring. So as with the plots, the tree is fully interactive. So if you were interested in selecting these ones, you can just click here. You can see that they're selected, or they're highlighted in blue in the table. You could extract them to a new table if you wanted to, or whatever you wanted to do with those. You can also change the distance of the tree. So if you wanted to drag, you can drag this, and the tree will adapt to the distance between each chemical. So we're going to leave it at 0.5. Okay, we can also change some different options. If you right-click on in the white space, you get some different options. You can close the clusters. You can open them. You can select centers. So this is going to select a center representation from each of the branches. Or you can be more specific and right-click and say select centers by, and you say by minimum, for example, molecular weight. Okay, that will select different centers. Or you can be more, you can add some additional parameters to that. You can say I want to select by minimum molecular weight, one from each cluster. But if I've got a very large branch with many compounds in it, I would choose a few more. It's a sort of some rules. So basically, if you have a larger branch, you would select slightly more than one for each, depending on how many there are. And then you may have singletons where there's just one member of a branch, and you can treat them as individual clusters or ignore them, whatever you want to do. So sometimes with like virtual screening or other data, you may want to use this, for example, for if you have a score or something, you could say I want to choose a diverse set of compounds with the minimum docking score from this list. This is a docking table. So just go OK. You can also right-click and you can save as an image, export it, and there's some preferences for you can change the way the tree is displayed if you want to. You can also use color to change the colors as well for the lines and other options, and line width and spacing and font as well. So here we have the name, the molecule name. You can also add additional data here. If you right click and say change record labels, so you have name. You may want to add the molecular weight to this so we can see the molecular weight. Okay. So basically what I did so far is right click on the white space and go change record labels, and then you can add molecular weight, or you say a pen. So now go here, we have the name as well as the molecular weight. You can also change, if we go back to change record labels, we can also add additional coloring to this tree. So you click on the use color option, and it'd be nice to see, it's like a heat map basically, so we can also display the molecular weight, log P, and any numerical column here. So I'm going to say set to all numerical. You see it's added this ICM command line language. It's basically selected all molecular weights, log P, polar surface area, molecular area, and then these ones are related to the actual tree. I'm not going to use those. And then you go OK. So now if we expand this, we have the coloring for like a heat map coloring for each of those parameters on the right hand side of the tree. So it could help in selections or whatever you see a useful way of visualizing the data. And if we had, when we'd made the tree, we can select by clicking on a node here, you can select certain things. But if when we made the tree we chose the option to calculate maximum common substructure, you'd also see the maximum common substructure of each branch by highlighting here, and it will show up. Okay. So finally, we're just going to end with maximum common substructure dendrogram. It's something like this. And so basically I read in that, so I delete these. So I read in the chemical dictionary, and then we have the chemical dictionary, which we were using just a minute ago, and then I went to chemistry, MCS, maximum common substructure dendrogram. Click here. It's asking you the table name and the column, how it's going to label it, and you can run this optimization of it's quite a lengthy thing, because you're comparing every single chemical to find a common substructure. So when you do it, it'll take a while to run with this example, but when it's done, you have this plot on the right hand side which you can zoom into, and you can see how certain drugs are. So we have the drugs in the chemicals in the green, and we have the common substructure shown in blue. So we can select here, takes us to you can make selections in this table, in this dendrogram as well. Or of course you can search for a substructure that you like using what we just been doing. So it's going to go default layout. So with the chemical search options we use, we can use the search tab again, and you can make a search query here for a substructure, and then find it in the table, and then find the chemicals that have similar common substructure. So if we do search and say select in source, it's going to select in our tree. If we search this dictionary, maximum common substructure tree, okay, search, and you can see that we have these. In a second, it takes a little while to search, and we can see that we have some selections here in red, because some here, and also in the table as well. And we can right click on here and say center on selection, and we can zoom in. I just lost the selection, but you get the idea. So when you zoomed in, you can see how these different drugs are related to a particular substructure, which is the common one. So you can also undisplay the name, or you can there's a variety of options to change the visualization in the dendrogram, but they're all here. You can uncheck if you don't want to show the name. You can just right click and choose uncheck. That will remove the name. You just have the substructure. Okay. And if there's a particular substructure you're interested in, you can make the selection of those chemicals around it, and it's in the table. Made a selection here. Okay, so thank you very much for your attention again. If you have any questions after the workshop, after the webinar, please feel free to email me. My email address is here, or call. In 2 weeks' time, we're going to be looking at chemical library enumeration. So that will include enumeration by Marcouche structure, R groups, enumeration of libraries via reaction, and then how to filter the results and look at multiple parameter optimization. And then the final webinar in this series is on structure-activity relationship analysis, and that's going to be presented by Eugene Rausch, who's our principal developer here at Molsoft. And he'll be looking at R group decomposition by scaffold, 2D structure-activity relationship analysis, matched pair analysis, and some other methods for looking at SAR data, as well as how to look at 2D bioisosteres as well. So that's in a couple of weeks. These two in next month. Okay, so thank you very much for your time, and appreciate your questions. I'll have a look at some of them now. Okay, so let's get back to this. Right, so you can read in larger molecules, but such as peptides, and they will be displayed in 2D as well. Yeah, you can read in whatever, but it's just visualization-wise, you need to make the cell bigger if you've got a very large molecule. But I don't have an example at the moment with that. Is there a limit to the number of rows and the number of columns? Well, basically, you're limited. The main limitation is related to the memory of your computer. So these files can be very large, and SDF files in particular, you could obviously go up to millions of compounds. So it depends on the memory of your computer how large the database you are looking at you can read in. But we have ways around that, and one of them is instead of reading in the 2D structures, if you've got millions of compounds, you can go to File, then Open, and drop down here, and then it says, so we have SDF format, which is the regular format, but you can also choose SDF as Smiles, which will enable you to read in millions of compounds, and it won't affect the memory of your computer.
And oh yeah, so MDL, yeah, I'm actually highlighting it here. So it's MDL as smiles, and so you can read in many millions of compounds. So I can just show you that in the manual. Actually, there's an example in the tutorial section of the manual called, in the Chem Informatics, I think, and it's called Working with Large Chemical Space. So yeah, so you can have millions of compounds. It'll come up in a minute, I think. It will have the smile string rather than reading in the full. You'll visualize the 2D sketch, but it's actually reading just the smiles string here. So that's the tutorial how to work with very large data in the graphical user interface. And actually, everything in the GUI is fully scriptable, so everything can be in the terminal window. Everything we've done has a command line, so you can run it on the command line there as well. Oh, is there a way to wash the library? That's a good question. Yeah. Yeah, so wash the library maybe to make a standard format to, for example, to get rid of salts, for example, was the question. So if you want to do that, if you have a chemical table, you go to chemistry, and then there's an option called standardize, and this will give you the option to remove salts, remove explicit hydrogens, and other standardizing things that keep. You can also keep the salt information in an extra column if you don't want to delete it. So yeah, you can do that on the local table, or you can do that on a file as well that's saved on your local machine. Had a question about how to change the Boolean from AND to OR. I guess that was in the searching. So here we had conditions, and you can change the Boolean here to OR or AND. So you can use this blank and just use these as well. If you wanted to search numerical data rather than chemical data, you can use this here, these options, and just double click here to change the Boolean. Copy and paste. Okay. Yeah. So I had a question about copying and paste. Yeah, if you had, I'm just going to clone this. So for data, you can copy and paste rows by right-clicking, selecting them, and right-clicking and copy and paste. But if you have blank rows, you can insert empty before. You should be able to just copy cell. Copy to the mic and then paste it here. And these were automatically calculated on the fly, the properties. But yeah, it'd be nice if it was just like Excel, right? I need to get an example. So column function new string. So you should just be able to. You should be able to make a selection and copy and paste between the two. But if you're adding full columns, it's best just to go to Tools, Table, and then append. You can append rows or add external columns. Use that rather than, because if you've got a very large table, you're copying it into the memory of your computer. So it's better to use the tools here to copy and paste. there's some other functionality of copy and paste which I need to get back to you on because I'm not 100% sure. So yeah, I'll double check. You had not about the dendrogram. Can the dendrogram only display the substructure and not the compounds? Yes, so basically you can make selections in the table and just so the substructures are at the end, or the substructures are highlighted in blue. So I think if we select all rows marked green, yeah, basically make a selection of the ones you don't want to display, and then you can uncheck here, uncheck visible, and then they'll become. So if we zoomed in here, you can select. You can just display. So it's a selection issue, a problem. I need to be able to. Yeah, just. I need to find out the exact way of selecting only the ones that are substructure and not the chemical. But you can definitely do that by using these visible show names and what have you here. Okay. Yeah, we had a question about protonation. You can enumerate the protonation state based on our model defined pH. Here you go to chemistry and then enumerate formal charge states here. Had another question about the dendrogram. I'll have to get back to you about that one as well. So is there a way to... Yeah, so we actually covered that last time, the question about conversion of chemicals. So if you read it in the smiles, we showed, if you probably didn't see it last time, but you can convert from, if you had a column of smiles read in from Excel, you can go to convert smiles to 2D, and that would convert it. Okay. And yeah, some other questions. I think I have to get back to you. Oh, we had a question. We actually covered that in a different. Actually, next week I think. Oh, yeah, multiple parameter optimization. So I had a question about things like blood-brain barrier and Lipinski rules and how to automatically assign a score. So this is called multiple parameter optimization. We'll cover that next week, or in 2 weeks' time. So I will show you that. So you get something like this. So you get this is it's not CNS, but we do have a one for CNS. So it's showing the molecular weights, number of hydrogen bond donors, hydrogen bond acceptors plotted in the table. So if you have, yeah, that's more of a another question. Yeah, I'll get back to you on some of these. I'll get back to you. That's multiple parameter optimization. We had a question about searching Kemble, because last time it's hard to run it because it takes a few minutes to run, so it's not ideal for a webinar. So if you go to search Kemble and then you can sketch the search query, or you can add text and that will return a table, or you can search by uniproduct, substructure, by smile string, by chemical name. That's in the search window. Okay, question about the product. So this is called ICM Chemist, but if you have ICM Pro, which is the desktop modeling package with docking and homology modeling, these tools, if you have ICM Pro plus virtual ligand screening, then the chemistry, everything you see here, is incorporated in that. Yeah, so you should have that. Okay, I think I'll have to get back to you by email, and really thank you very much for your time. Appreciate your support of the webinars. Thank you.